So we move to the following speaker. Who is Fumihito Arai, a professor in the Department of Bioengineering and Mechanical Engineering at the University of Tokyo. And he will talk also on micro and nano robotics, manipulation and automation at small scales in the biomedical field. The floor is yours.
Okay, so Karume, thank you very much for kind introduction. My name is Fumihito Arai from the University of Tokyo. Today, I will talk about micro and nano robotics, but I may not talk about a small robot, but I will talk about manipulation and automation at small scales. Okay, so first of all, let me mention about market of global lab automation. It is growing like this, okay? So how can we contribute to the lab automation? So robotics and automation is important as the labor force decreases in the future. And of course, AI is expected to contribute to scientific discovery and is well suited for solving such problems. And in case of lab automation, there are so many demands such as high throughput, high accuracy, or something like that. So in terms of high accuracy manipulation, I think micro and nano robotics will also contribute to this area. So today, I may focus on, especially on scientific experiments, like for drug discovery or something like that, okay? So manipulation object for scientific experiment, for example, of course, equipment and tools, and of course, living things and components, right? The size ranging from 1 nanometer to centimeter order. So the scale gap is 10 to 8. It's a very multi-scale object, okay? So, the role of the robotic system is specified, especially in a case of biomedical works. I may roughly classify into two cases. One is analytic use, and another is experiment use. Another word, chip-based approach or test tube-based approach, okay? And in case of manipulating small object, it is very difficult, of course. The size ranging from 1 micrometer to a centimeter order, like a cell and oocyte, right? And, tissue, of course. And, for the last 30 years or 40 years, micro and nano robotics focused on manipulation on small scales, okay? So in case of small-scale environments, the effects of disturbances are more influential. So disturbance makes manipulation very difficult. However, we may utilize lab-on-a-chip. It is a quite useful device that integrates laboratory functions on a small microfluidic chip, okay? And, to expand the manipulation capability at small scales, I think microfluidics is a very powerful tool. For example, environmental control is possible, and especially elimination of disturbances is possible, right? And of course, robotics. So individual manipulation of small objects like cell is possible with high speed, with high spatial resolution. Okay, so why not integrate those two different technology, microfluidic chip technology and robotics, okay? So to do this, I may classify into two ideas. One is robot on chip, and the second one is a chip on robot. The first case, the chip is fixed, right, under the microscope, and robot access to the target place, okay? So sometimes the robot will be miniaturized and put inside the closed space. Then we need a non-contact drive manipulation method. I used optical force and magnetic force as shown here. And of course, another case is contact drive. We may make a small miniaturized structure using MEMS technology that can be driven by some precise actuation technology. So contribution of robotics is here, manipulation of small object, of course, and of course, also we can control the flow in the microchannel. Okay, so manipulation capabilities are expanded at small scales in the last 20 years, I think. So one is liquid manipulation. We can mix the liquid in the channel in high speed, less than 1 millisecond, and solution exchange and flow switching in more than 20 kilohertz order. And another one is manipulation of cells and the tissues, and we can measure the force and the stiffness variation is possible. Like this, okay? So let me mention one of our example of our innovation in liquid biopsy. We have developed a rare cell sorter, which can isolate cancer cell. It is a circulating tumor cell, CTC, in the blood sample. And we can correct the separated, isolated single CTC by robotic technology, like this system. And we transported this technology to the university startup, Medridge. Well, anyway, so this system employs a new separation method using a controlled air-liquid interface. And we realized damage-free filtering of rare cells, CTCs. And the important thing is that cancer diagnosis is possible with only 5 milliliter blood sample. So it is quite low invasive and less patient burden. Okay? So go back to the lab automation. What is a critical issue? As I told you, analytical use and experimental use, and currently the next generation sequencing technology is advanced. Then, well, handoff of small samples like cells is required, but it is very difficult, right? And reliable sample transfer with zero contamination at multi-scale level is still very difficult, okay? So I may say the contribution of micro and nano robotics in this field for the past 40 years, maybe analysis, manipulation, and automation at small scales. But look here, the size ranges is from nanometer level and centimeter or meter level, right? So volume ratio is 10 to 24. It's a multi-scale, right? So manipulation automation is very difficult. So I think the next challenge will be the analysis, manipulation, and automation at multi-scales, okay? So let's think about the example. This is a photograph of our laboratory in Tokyo. It's very small laboratory, but as you can see, work sites are dispersed, right? But we need to manage individual cell in the process. So the small cell travels from site 1, site 2, or something. And well, there are several issues like secure transport of small sample in the large laboratory. Really, it's small, but for the cell, it's very large. It's very difficult. And environment are extremely sensitive to the external disturbances, and individual management of samples is needed without contamination, okay? So key technologies of lab automation at the multi-scale, I may mention 3. One is, of course, dexterous manipulation. We realized the remote center of motion mechanism with less than 5 micrometer position accuracy. And the tracking of manipulation is possible using autofocusing, and it is working under the, well, for example, confocal microscopy. And last one is the individual management of cell. In this video, small cell flow is managed in a small microchannel, okay? Well, I may say this example more. As I told you, the combination, integration of the second case, chip on robot. In this case, chip is manipulated by the robot, so it can move around the laboratory, okay? Therefore, the individual management of cell without contamination will become possible. So I may show you one example, individual transport of oocyte. This is used for drug discovery. This is very important. And as you can see, a robot carries a chip with oocyte in it. And the chip loads the oocyte at one at a time, okay? So efficient transportation, contamination-free, of course, and individual management of cell is possible. Okay, let me summarize my talk. Today I talked about micro-nano robotics, especially focusing on manipulation and automation for the last 30 years. And integrated use of robotics as well as microfluidics provided new functionalities, right? And manipulation capabilities not only at the small scale, but also the multi-scale are becoming more and more important for lab automation. And of course, making robot autonomous involves many challenges. There are many good talks on AI robotics. So maybe the effective use of AI will be our future challenge. Okay, this is the end of my talk. Thank you very much.
So we have time for one or two quick questions.
Thank you very much.
You were so clear with the future that I don't see anyone asking questions. Well, to me, it was very clear that the multi-scale thing was very important, and also the incorporating AI in the next year, right? So I don't know if you like to add something to this, or we close here.
Yes, I have a question.
Rudiger, please.
Oh, thank you.
Okay.
Thank you for your talk. Only one question. Nanometer is extreme. How you can reach this accuracy? Is this absolute accuracy or relative accuracy? And what is the trick to be so precise?
Yeah, thank you very much. It's a critical issue. Actually, it depends on the actuation system and the observation system. So in the field of analysis, nanometer order RNA analysis is very popular. For example, single cell and RNA sequencing. But I think you are more thinking about the direct manipulation of small molecular level. In that case, I think observation is very important. So manipulation system with the observation system should be mixed to each other. In that sense, we have several other technology like a new high resolution imaging method and AFM, of course. But we'll still need real-time manipulation while observing in real time. It is still challenging, right? But we have to do more about a much more larger scale, cell level. So many things should be done. So today I focused on cell manipulation or such kind of automation issue. And environmental control is quite important. That will be also true for the nanometer level manipulation. Thank you very much.
Thank you.